The contraction of heart muscle in all animals is initiated by electrical impulses known as action potentials. The rate at which these impulses fire control the rate of cardiac contraction or the heart rate. The cells that create these rhythmical impulses are called pacemaker cells, and they directly control the heart rate. In humans, and occasionally in animals, a mechanical device called an artificial pacemaker may be used after damage to the body's intrinsic conduction system to produce these impulses synthetically. Control primary equals 1% of the cardiomyocytes in the myocardium possess the ability to generate electrical impulses spontaneously. A specialized portion of the heart, called the sinoatrial node, is responsible for atrial propagation of this potential. The sinoatrial node is a group of cells positioned on the wall of the right atrium, near the entrance of the superior vena cava. These cells are modified cardiomyocytes. They possess rudimentary contractile filaments, but contract relatively weakly compared to the cardiac contractile cells. The pacemaker cells are connected to neighboring contractile cells via gap junctions, which enable them to locally depolarize adjacent cells. Gap junctions allow the passage of positive cations from the depolarization of the pacemaker cell to adjacent contractile cells. This starts the depolarization and eventual action potential in contractile cells. Having cardiomyocytes connected via gap junctions allow all contractile cells of the heart to act in a coordinated fashion and contract as a unit, all the while being in sync with the pacemaker cells. This is the property that allows the pacemaker cells to control contraction in all other cardiomyocytes. Cells in the SA node spontaneously depolarize, ultimately resulting in contraction, approximately 100 times per minute. This native rate is constantly modified by the activity of sympathetic and parasympathetic nerve fibers via the autonomic nervous system, so that the average resting cardiac rate in adult humans is about 70 beats per minute. Because the sinoatrial node is responsible for the rest of the heart's electrical activity, it is sometimes called the primary pacemaker. Secondary if the SA node does not function properly and is unable to control the heart rate, a group of cells further down the heart will become the ectopic pacemaker of the heart. These cells form the atrioventricular node, which is an area between the left atrium and the right ventricle within the atrial septum, will take over the pacemaker responsibility. The cells of the AV node normally discharge at about 40 to 60 beats per minute, and are called the secondary pacemaker. Further down the electrical conducting system of the heart is the bundle of His. The left and right branches of this bundle, and the Purkinje fibers, will also produce a spontaneous action potential at a rate of 30 to 40 beats per minute, so if the SA and AV node both do not function, these cells can become pacemakers. It is important to realize that these cells will be initiating action potentials and contraction at a much lower rate than the primary or secondary pacemaker cells. The SA node controls the rate of contraction for the entire heart muscle because its cells have the quickest rate of spontaneous depolarization, thus they initiate action potentials the quickest. The action potential generated by the SA node passes down the electrical conduction system of the heart, and depolarizes the other potential pacemaker cells to initiate action potentials before these other cells have had a chance to generate their own spontaneous action potential. Thus they contract and propagate electrical impulses to the pace set by the cells of the SA node. This is the normal conduction of electrical activity in the heart. Generation of action potentials. There are three main stages in the generation of an action potential in a pacemaker cell. Since the stages are analogous to contraction of cardiac muscle cells, they have the same naming system. This can lead to some confusion. There is no phase 1 or 2, just phases 0, 3, and 4. Phase 4, pacemaker potential equals. The key to the rhythmic firing of pacemaker cells is that, unlike other neurons in the body, these cells will slowly depolarize by themselves and do not need any outside innervation from the autonomic nervous system to fire action potentials. As in all other cells, the resting potential of a pacemaker cell is caused by a continuous outflow or leak of potassium ions through ion channel proteins in the membrane that surrounds the cells. The difference in pacemaker cells is that this potassium permeability decreases as time goes on, partly causing the slow depolarization. Also there is a slow, continuous inward flow of sodium, called the funny current. These two relative ion concentration changes slowly depolarize the inside membrane potential of the cell, giving these cells their pacemaker potential. When the membrane potential gets depolarized to about -40 MV, it has reached threshold, allowing an action potential to be generated. Phase 0, upstroke equals, though much faster than the depolarization caused by the funny current and decrease in potassium permeability above, the upstroke in a pacemaker cell is slow compared to that in an axon. The SA and AV node do not have fast sodium channels like neurons, and the depolarization is mainly caused by a slow influx of calcium ions. The calcium enters the cell via voltage-sensitive calcium channels that open when the threshold is reached. This calcium influx produces the rising phase of the action potential, which results in the reversal of membrane potential to about +10 MV, where it peaks. It is important to note that intracellular calcium causes the muscular contraction in contractile cells, and is the effector ion. In heart pacemaker cells, phase 0 depends on the activation of L-type calcium channels instead of the activation of voltage-gated fast sodium channels, which are responsible for initiating action potentials in contractile cells. For this reason, the pacemaker action potential rising phase slope is more gradual than that of the contractile cell. 7. Phase 3, repolarization equals, the reversal of membrane potential triggers the opening of potassium leak channels, resulting in the rapid loss of potassium ions from the inside of the cell, causing repolarization. The calcium channels are also inactivated, soon after they open. Also sodium permeability into the cell becomes decreased as the sodium channels become inactivated. These ion concentration changes across the membrane slowly repolarize the cell to resting membrane potential. Another important note at this phase is that ionic pumps restore ion concentrations to pre-action potential status. The sodium-calcium exchanger ionic pump works to pump calcium out of the intracellular space, thus effectively relaxing the cell. The sodium-potassium pump restores ion concentrations of sodium and potassium ions by pumping sodium out of the cell and pumping potassium into the cell. Restoring these ion concentrations is vital because it enables the cell to reset itself and enables it to repeat the process of spontaneous depolarization leading to activation of an action potential. See also, cardiac action potential, electrical conduction system of the heart, artificial pacemaker. References